Hello, everybody, and thank you very much for joining us this afternoon, or this morning, or this evening, depending on where you're calling in from today. My name is Louise Tratt, and on behalf of the British Neuroscience Association, I'd like to give you a warm welcome to this special webinar, Introduction to Multicell Patch Clamp Recordings, which, of course, is hosted jointly with our annual supporters at Scientifica. Just before we start, and whilst people are still joining, we have just a few housekeeping rules to go over. We'll be using the chat and the Q&A function today, so please do feel free to say hello, post comments in the chat, let us know where you're calling in from today. If you have any specific questions for our speakers, then please make sure you use the Q&A function for these. And you should see both the chat and the Q&A buttons at the bottom of your screen. There'll be a Q&A session after the talks with a chance for you to ask our panelists any questions and that you may have. So feel free to post your questions at any time during the talks and we'll pick them up at the end of the session. You'll have the option to ask these questions anonymously if you wish to do so. I'd like to take this opportunity to remind you this webinar is being recorded with the permission of our speakers and it will be available to watch on demand on the Scientifica website and the BNA YouTube channel. And finally, just a reminder to please adhere to the BNA Code of Conduct for webinars and engage respectfully online. So, without further ado, it gives me great pleasure to hand over to Dr. Max Oginsky, Product Specialist at Scientifica, who will officially start the webinar today.
Thank you. Thank you for joining our webinar here for multicell patch clamp electrophysiology. I'm going to begin by giving an overview of patch clamp electrophysiology, then move on to who may benefit from multicell patching, and then kind of give an overview of the technique and how I might go about doing it, and then incorporating the benefit of using scientific systems when you're doing your multicell patching. Patch clamp electrophysiology, we know, can be done on many excitable membranes or cells, but for the day today, we're going to talk about neurons basically and patching from brain slices. So you can visualize a brain slice under a microscope. Here's the Scientifica Slice Scope Pro 6000, where we have our microscopes. You can visualize your neurons, and then you can use a patch jar micromanipulator to move the electrode and pipette to towards your cell. Once you bind to the cell, you can once you touch the cell, you can make a giga-ohm seal, then use a reverse pressure to gain electrical access, so you have this whole cell configuration. I know when I started patching, I started recording action potentials, so you patch onto a cell, give a depolarizing current injection, and then you can record the response of the neuron. In this case, we have a train of action potentials. And from this, you can measure a lot of different facets of the activity of the neuron, which we call the intrinsic membrane properties. And then over the years, people have been able to do experiments that figure out which ionic currents underline these different these intrinsic membrane properties. So for instance, we know that the inward rectifying potassium current underlies the resting membrane potential, voltage gated sodium currents, regulate the threshold and amplitude, etc. And so what's really great about this technique, besides a lot of things, we can, any kind of drug application, you can find the mechanism of action just by looking at changes in the shape of the action potential. And then you can know, based on past work, what's the likely candidate the drug is, what current or channel that drug is acting on. If you're more interested in how neurons communicate with each other, you can look at evoked postsynaptic currents. In this case, this is an AMPA current, so it's excitatory. You can stick a stimulating electrode into your slice and inject current into the slice and then record this postsynaptic current. So in this case, you can see that the insulin is causing the EPSC to increase. The problem is you can't really see what the mechanism is. Is it presynaptic versus postsynaptic? So you can, to get at that answer, you can record miniature postsynaptic currents. Miniature is just like a action potential independent measure. So these downward deflections are what we call minis. And then if you see an increase in the frequency or a decrease, you can kind of infer that that's a presynaptic mechanism. Changes in amplitude are usually considered postsynaptic. And then you can use a paired pulse ratio to verify that those findings in the minis. Paired pulse ratio is just kind of a measure of the release probability. So these are evoked postsynaptic currents again. So you put them close together, and if the ratio changes, it indicates more of a presynaptic mechanism, whereas if they don't change, more of a postsynaptic mechanism. The problem with electrical stimulation is that you're not able to identify the neurons that are actually synapsing onto your postsynaptic neuron, the neuron you're recording from. Obviously, with the advent of optogenetics, people have been able to express channel rhodopsin into specific neuron types, and then by shining blue light, you can elicit a EPSC, and then kind of look at the connectivity that way. So here's like a diagram where that might be pretty useful in the past. Here's a cortical column with an excitatory neuron, and then you have a number of inhibitory neurons. So people have done experiments where they express channel rhodopsin in these different types of inhibitory neurons, and then looked at the connectivity to this excitatory neuron here. Now, this is a simplified diagram. You don't really know what neurons are, what these inhibitory neurons, how they're communicating with each other, but they do a synapse onto each other. And this is where a multi-cell patching approach may be very beneficial. We have an article on the Scientifica website, which you can see here by this link. This is a figure in that article. And so what we see are 4 cells that are patched in a layer 5 neuron in the neocortex. You can see these neurons using contrast. And if you look at this figure, each of these cells are firing action potentials here. And then you can see the response of the other neurons below it. So for instance, what we see is when cell 1 is firing an action potential, cell 2 here is responding with these EPSPs. When cell 2 is firing action potentials, you can see that cell 3 is actually being inhibited. Here you can tell by the IPSPs. So this is really great because not only can you figure out the connectivity, but based on the response of the postsynaptic neuron, you can also identify what that type of neuron, that presynaptic neuron is. One of the disadvantages of this is that, in the cortex or in other areas, the connectivity can be sparse. So if you're doing paired recordings, it may be really difficult to find neurons that are connected. But as you increase the number of cells that you're patching, those possible connections grows. So for instance, 4 cells, you can look at a possible 12 connections, 5 cells up to 20 possibly, unlikely of course, but it gives you that ability, and then 6 cells could be up to 30 connections. And the reason that I'm telling you this is not because I want to give you a history lesson on patch clamping, but because so much work has been done, and we found out so much just by patching onto single neurons, that I really think that this multi-cell approach can really improve data production, efficiency, and really bring us a lot of new answers to some questions that have been out there. So who else may benefit from multi-cell patching? Anyone who's interested in neuronal connectivity within these networks. So basically if you're interested in how neurons influence each other's firing, how experience affects the activity and connectivity of changes. I know a lot of people do train mice or rats to perform a task to obtain a reward like cocaine or alcohol or sugar. So after you train that mouse, you can kind of look at those changes within a small network. Others that may be interested are people that do studies with aging or use age-related disease models. Sometimes these aging studies last, they house the mice for upwards of a year or more. So it's a lot of money, a lot of time. And so you want to get the most bang for your buck, right? So if you're able to do multi-cell patching, it's really improves your efficiency and really helps you out in that case. If you're a modeler, you know, I know reviewers really like more biologically relevant models. A lot of models are single neurons, but by using multi-cell patching, you can build a larger model and have more biologically relevant data. There are models that have thousands of neurons and they have this connectivity probability within them. Obviously, all 1,000 neurons are not connected, but they incorporate this probability into their code. And so if you're able to do these recordings, they have more of a accurate probability there. And you know, larger data sets improve models. I think one of the things with electrophysiology in general is having, there are differences from one lab to another. There are differences from one patcher to another, even one animal to another, right? So we have these little differences that can make, you know, bring in all this, a real understanding of what's happening, making that more difficult. So as a modeler, so if you are able to get this kind of information, this kind of data, it just makes your models that much better, I think. And lastly, I think one of the big picture questions in neuroscience is how an animal perceives its environment. Then based on that sensory information, makes a decision and then makes a plan of action. So we have customers who do multi-photon imaging. They train a mouse to perform a task, and then they image the activity of those neurons. And then after the mouse learns the task, they remove the brain, do patch clamp recordings, and really kind of find out the nitty gritty of what's happening there. And so you can, I think that multi-cell patching, again, as a way of being as efficient as possible, is a really powerful technique. So once you do those experiments, you want to be able to get as much data as possible. So moving on to how I would go about doing multi-cell patching, I say the steps I would use because you know, I think there are small differences between, you know, one might have to consider depending on your experimental needs. I do think automation is a really helpful way, which Scientifica has an excellent package to be able to make things easier on the patcher. So to begin with, just like any other electrophysiology experiment, you need to find your healthy cells. And then with Scientifica, we have the LinLab 2 software. So within that software, we have this store points function, which is this icon right here. And so what we do is we can click on the stored points function. This window is going to pop up. And you have these 3 tabs, this MMTP, which is our motorized movable top plate, which you see in this picture here, right? And then you can just click add on the location. So once you find a cell that you like, you just click add, it'll pop up in this window. And then later on, when you click on that cell again, the top plate will move right to that spot. So once you go through all of your cells and you find like 4, 6, 8, maybe cells that you want to patch, then you can move your pipette tips under the objective, okay? I would make a gigaohm seal on the first cell, usually find the deepest one of the cells that you want to patch, and then move to the next cell, make a seal, and just repeat this as needed. And once you make a seal on each cell, then you can break into each cell. Now, I do know a lot of people who, when they're doing these experiments, they just make a seal, break in, and then move on to the next cell. But I think this would be my approach. And then after you break into each cell, you can start your experiment. Now, one of the things I want to focus on is actually the moving of the pipettes once you make your seal on your first cell and then moving to the next cell. So we have another part of our LinLab software called the follow function. And so what this does is it allows you to link the manipulators, these 2 manipulators, to to the stage, all right? And so what this does is it keeps the pipettes in the field of view. And so then with those stored points that I just talked about earlier, you can double click on each point and then move the stage to that point while your pipettes are under the field of view. And so when cells are farther away from each other, this makes those recordings a lot easier. So a quick way to do that, you can either click on the follow button here, or you can click on this icon here, which is two pipettes that are close together. When you do that, this follow window pops up. This motorized movable top plate is called the master device, and then you can click on here, and so that the patch stars are following the top plate there. So here's just a picture of two pipettes. Once the pipette on the left makes a seal onto this cell, then what you can do is click off, and then this pipette on the left will stay while the other pipette is able to keep moving. And so this makes everything a lot easier. It's just fully automated in that sense. And to move the pipette from one spot to the next, or from one cell to the next, you can just double click on that stored point. We have, as part of our micro manipulators, we have the patch star and the microstar. Both are highly stable. The low drift with less than a micron for 2 hours. So it does take a long time depending on how many cells you're trying to patch. These could take a while. It's very heavy, so it really keeps your pipette in place. If you want to do a lot of cells, maybe, we have the ability to go up to 12 microstars around a dish if you have an extended motorized movable top plate. But as you can see here, it has a very slim design. So depending on your experimental needs, you could fit a lot around your dish. And this sliding carriage makes it really easy to change a pipette. And this, I forgot to mention, on the patch star, you have this ability to rotate along this axis. So even though you have maybe have a few patch stars around your dish, it's really easy to exchange your pipette just by rotating this out of the way of the dish. Here's a picture of another customer, Ethan Goldberg at UPenn. And you can see 4 patch stars in the back and then 2 microstars in the front. So you are able to combine them together. They both work with the LinLab software the same. It's really a matter of preference in that case. So to summarize everything, I think the benefits for researchers interested in networks, if you're really interested in how the brain models our environment, predicts the effects of actions, and how this is done within networks. If you want to be more efficient, more productive, this is a powerful technique. It's a lot like single cell patching, but you need to take extra care so that these pipettes don't bump into each other, and that might take a little longer. But the automation that we provide at Scientifica with our system limits these mistakes. So really, the integration of the components, the patch stars, the staging, along with the LinLab software, really allows our customers to really enhance their efficiency. And if you have any more questions on electrophysiology, you can always visit the Scientifica website. You can see a link down here. And here's some titles to some articles that I thought would be helpful. And with that, I'd like to introduce Stephanie Seeman, who's a researcher at Allen Institute. She has some really exciting data, and I'm excited to hear her story.
Yeah, thanks, Max. I will start. Oh, I think, Max, you need to stop sharing before it will let me share.
There we go.
Can you stop sharing your screen real quick, Max? Oh, there we go. All right. Great. Hopefully everybody can see my screen now. Yeah, so thanks, Max, for the introduction and for the invitation to talk to you all about how we at the Allen Institute leveraged the multi-patch technique and Scientifica devices. I've been a scientist there for the last 6 or so years. And during that time, we undertook a very large project to explore connectivity and synaptic properties in mouse and human cortical slices using multi-patch. So hopefully we can all agree and are familiar with the idea that the brain is composed of multiple cell types. As Max alluded to, we have excitatory pyramidal cells and inhibitory interneurons, and these cells can be identified by their morphologies or the intrinsic electrical properties that can be recorded. But they also have unique and specific connectivity that exists between them. And for those cells that are connected, the properties of those synapses, or what we call the synaptic dynamics, are also influenced by the identity of the pre and postsynaptic cell type. And so we were interested in exploring this at a large scale. These ideas are not new. People have been studying the connectivity and synaptic properties of different cell types for almost a century. But we saw an opportunity to do this at a large scale using multi-patch systems. And so just to sort of reiterate what Max said, if we can record from multiple cells at once, in turn, we can stimulate one cell and drive it to elicit action potentials. And then while recording from our other cell, see the, in this case, excitatory postsynaptic potentials in the connected cells. And when we only have 2 cells to record from, as Max mentioned, it's fairly low throughput. We can only explore the 2 connections that exist between these 2 cells. But we bumped it up to 8. And by doing that, we can probe up to 56 potential connections. And so by doing this, really increases our throughput and allows us to explore the local connectivity more thoroughly. So we built 5 octopatch, what we call octopatch rigs at the Allen Institute, and just wanted to show off some pretty pictures of our systems. Here you can see our recording chamber here in the middle with 8 augmented patch stars oriented in a semicircular fashion around the chamber. And here we see one of our research associates who did many of these recordings sitting at his rig with a suite of monitors and tools to do these experiments. So while we were building these systems, we learned a lot of things. And so to kind of add on to what Max talked about and how he would go about these, we learned a lot of things along the way that I think can be helpful to any of you who are looking to set up a system like this yourself. And as Max has already mentioned, automation is one of those key elements that is necessary to do these experiments efficiently. It's really hard once you've gotten up to 6, 8 electrodes to keep track of them all. So tracking the movement either with the LinLab software, or we took the LinLab software a little bit further and added to it with some of our own automations to reduce clicking and have everything be controlled by one board. But any way that you can know where your pipette tips are, know where your target cells are, and have things move in an automated way will save you a lot of time and headache. Something else that we automated was the pressure regulation. So as you're either approaching the cell or then sealing onto the cell, you want to have positive or negative pressure, and that needs to be switched sort of instantaneously. And one of the more traditional approaches is to use a manual mouth pressure regulation. But again, it's hard to control 8 independent pressure channels with your mouth, unless you're some sort of crazy musician who can do those sorts of things. And so we used automated pressure regulation system to dynamically switch pressures on the various head stages and electrodes as was necessary. And then finally, once you've got all of the cells that you are recording from, this is precious time, and holding all of those cells won't last forever. So you want to be able to go through your experiment as quickly and efficiently as possible. So we spent a lot of time honing our stimulus protocol so that with basically the press of a button, the experiment would run itself while also checking that cells were staying healthy and were still spiking and these sorts of things, so that we could repeat stimuli as necessary. And it really required minimal intervention by the experimenter to keep these experiments on track and have them be as complete as possible. And so, as has already been mentioned, we're packing a lot of things into a small space here. And we were a bit greedy, and we wanted both the stability of the patch stars and the slim profile of the microstars. So we augmented these a bit. Hopefully you can maybe see here. Using the patch stars with their stacked motors, we also then took the head stage off of the side where it normally resides and mounted it right on top of the top motor here, so that everything is a nice slim column, and we could pack 8 of these as close as possible. And we did that because we noticed that when you've got electrodes or motors stationed further out on the slice scope, there's a bit of physical, we call physical crosstalk or vibration. So when this pipette moved, you can see that some of these other ones are vibrating, some more violently than others. And you can imagine that if those electrodes are patched onto a cell, the likelihood that you've still got your cell is pretty small. So we did a lot of work to try and push those motors and manipulators as far into the center of the stage as possible, while also working a lot with Scientifica to really reduce the magnitude of those acceleration induced movements. And then one last piece of optimization was eliminating electrical crosstalk. So when you've evoked an action potential on one electrode, it's not uncommon that in an adjacent electrode, you see an artifact of that, that action potential. And this can be problematic down the line, because if you imagine that there might be a connection between these two cells that you want to see, that connection is now being overshadowed by this electrical artifact. It's now buried somewhere in here that you can't resolve. And so we came up with a fairly simple, but I think elegant solution to this, where we manufactured these aluminum shields, bent them in a few places, and then clipped them onto the side of the pipette holder, so that now there's a electrical shield in between pipettes. And you can hopefully see in this trace that between pipettes that had a shield, the electrical artifact is practically eliminated. So these are just sort of the main facets that we optimized while we were building this rig and all of these rigs. But that still doesn't mean that there weren't other things along the way, and these are still incredibly hard experiments. And so even when we did still get 8 simultaneous recordings, we wanted to celebrate that, and we would activate our octo alarm. And so this is just a fun thing that we did and wanted to share with you all. Here you can see that we would turn on our little alarm in the lab, and everybody would come running, and it was usually a great day. And another excuse to show off a really nice recording here with 8 recorded cells. In the diagonal is where you can see each cell being driven to fire an action potential. And then if you look up and down a column, then you can see evidence of synaptic connections. So like this blue cell, when it fires an action potential, you can see a small EPSP in the orange cell, or this yellow cell here induces a large response in this gray cell. And so this is kind of what our data looks like coming off of the rigs. And then we spent a lot of time analyzing it and figuring out just what was going on between all of these recorded cells across many experiments. So with that, I'll just want to highlight very high level some of the data and results that we found over the course of this 6-year project. So our main goal, as I mentioned at the beginning, was to investigate the cortical connectivity in this case, mouse primary visual cortex and human frontotemporal cortex. We primarily were looking at chemical synaptic transmission between cells, but often we would find electrical synapses or gap junctions, particularly between inhibitory interneurons. From these chemical synapses, we characterize properties such as strength and kinetics and short-term plasticity, as well as formed a model of synaptic types that we hope will be useful to the modeling community. And so these this set of experiments was very high throughput, and we think is one of the largest data sets that exists on this. And while many of these things have been done in previous research operations, we feel that this is one of the first places where all of these data exist at once and were acquired under a standardized protocol so that people can really get a holistic view of local cortical connectivity. And at a very high level, some of the things that we learned from these data were that the connection probability does vary between layer. So we found that in superficial layers, such as layer 2, 3, there was a generally higher level of connectivity than in some of the deeper layers, such as layer 5. And in terms of synaptic properties, these also had characteristics that were dependent on different cell types. So in general, we saw that excitatory synapses were more influenced by the postsynaptic cell type, while inhibitory dynamics were more influenced by the presynaptic cell type. And one of the main drivers of these synaptic dynamic differences was something we call synaptic variance. And so I'll go into that a little bit in a few slides. So first we can explore our connectivity results. Here showing you 2 connectivity matrices, our mouse connectivity matrix on the left and our human connectivity matrix on the right. And if we focus first on the mouse connectivity matrix, in these we have presynaptic cell types along the rows and postsynaptic cell types along the columns. Max had mentioned using oncogenetic techniques to know the identity of pre and postsynaptic cell types. We took a transgenic approach. So we bred mice that were quadruple transgenic in that they expressed in usually TD tomato in one cell type and a GFP in a second cell type. And in this way, we could explore this whole matrix in a sort of pairwise fashion of 2 cell types at a time. And we mostly focused on connections within a single layer. So these white lines are denoting each of the layers. And so you can kind of see that these boxes along the main diagonal are our intralaminar connectivity. And that's where we performed most of our experiments, but got some interesting off-diagonal or intralaminar connectivity as kind of an aside when we didn't know exactly what layer we're in. And so in these matrices, the warmer the color, yellows, the higher the connectivity, and cool colors, blues and purples, are lower connectivity. And there's a lot of interesting things to look at in here, but hopefully you can appreciate what I mentioned before, and that the upper layers, layer 2, 3 here in particular, just has a lot more yellow squares, higher connectivity than the layer 5 box here, which is more blues and purples. And this was mirrored in our human data. So while in the mouse, we could use these transgenics to target excitatory and inhibitory cell types, we don't have transgenic humans yet, so we had to rely on identifying cells mostly by morphology, and so pyramidal cells are the easiest cell type to pick out. So here we are mostly looking at excitatory pyramidal cells in each of the layers of human cortex. And again, in this expanded layer 2-3 of human cortex, we see higher connectivity than in the deeper layers 4 and layers 5. And what was most striking is when we compared layer 4 between mouse and human. So connectivity among layer 4 pyramidal cells in humans was extremely sparse, in particular compared to the connectivity among pyramidal cells in layer 4 of mouse, which was actually one of the higher connection probabilities that we saw. And so that warrants further investigation and has, potential interesting consequences for cortical circuits and function if we consider that layer 4 is typically the input layer to different cortical regions. And so along with the color scale here, you might have noticed that some of these elements or squares are darker in saturation than others. So like this layer 4 box again has a dark blue color, whereas some of these others are lighter blue. And we used this saturation to impart our confidence interval on the measurement, the connectivity measurement here. So for an element like this, where we recorded from 145 pairs, we're more confident that this connection probability is accurate compared to an element, say, that we only recorded from 6 pairs, in which the fact that we didn't find any is more that we didn't probe it hard enough, rather than that these two cell types aren't connected to one another. So aside from a throughput, there are other technical reasons why we might miss connections. And so in slice electrophysiology, we consider these connectivity measurements a lower bound to connectivity. And these, one of the things that affects that measurement of connectivity the most is signal to noise ratio. So here I'm just showing you 3 examples of background noise in 3 different recordings, and hopefully you can appreciate that this cell on the left has a lot of background noise. It spans about 10 millivolts, and one can imagine that if a small millivolt or sub-millivolt response is buried in there, it's going to be hard to see, especially compared to a really nice recording like this one, where the noise is very small. And things that contribute to noise can be, there are many things that can contribute to noise, and so narrowing those down, while important, you're going to have to deal with this to some extent no matter what. So to get around this, or to sort of counteract it, one way is to make sure that you have a lot of trials of your stimulus. So here showing examples where if you only had 10 trials probing a pair, giving a presynaptic spike in the presynaptic cell and recording a response window in the postsynaptic cell, you can see that it appears that there's no connection here. Even when we've delivered 50 presynaptic spikes, yeah, maybe there's a little hump there, but it's pretty noisy. I don't know that I would trust that 100%. But once we get up to hundreds, almost thousands of trials or presynaptic spikes, now we can start to see quite clearly this small synaptic response buried in this large noise. And we can measure these attributes, the background noise and the number of trials, for every pair that we recorded, and look at how that relationship is with the connection probability. And so we combined these 2 individual metrics into what we call detection power, which is analogous to a signal to noise ratio. And you can see that when detection power is low, either the background noise is high or the experiment terminated early and we didn't get enough trials, that our ability to detect connections is very low. However, when detection power is high, we can more reliably detect connections. And so by measuring these things in all of our data, we can know where on this curve all of our data lie, and thus adjust our confidence in the connection probability, knowing this relationship. And so that was one aspect of our analysis that we think was really critical for moving forward in this field, because it's hard in prior experiments to really compare connection probabilities across experiments and across labs. And so by quantifying these relationships and allowing folks to place their data within this regime, we can sort of normalize things and have better comparisons across data sets. So once we found connections, we wanted to characterize their synaptic properties. And here you can see one connection where we've got many individual PSP trials in the gray. We can average those all together to create this average trace in the blue. And then we would fit that average trace and extract our synaptic parameters from that fit. Things such as the latency or the amplitude or strength, rise time, decay time constant, and those sorts of things. We could then use these fits to go back to the individual PSPs and use the fit parameters there as a starting point to fit individual PSPs. And then by doing that, we could look at how individual responses varied from trial to trial and over time in an attempt to quantify things such as short-term plasticity and this variance metric that I alluded to earlier. So here, looking at a train of responses, we can see that within each pulse, the variance from trial to trial, exists, but that overall, the response to this first spike in the train is generally larger than the response to the last few spikes in the train. And so this type of synapse would exhibit short-term plasticity depression, as opposed to facilitation, where the response would grow over time. And so similarly to our connectivity matrices, we can look at these in a matrix form, again, with presynaptic cells on the rows and postsynaptic cells on the columns, and colorize these matrices by whatever synaptic property we are interested in. And so some things can immediately stand out from these matrices, such as parvalbumin interneuron connections having a fast latency. They're also very depressing. If we look at our short-term plasticity matrix here, they have a strong blue color. We can look at things like rise time, amplitude, among many others. Once we start to get up to, multiple properties that we're interested in, it becomes a little hard to look at these individually and understand how they interact with each other. And so to get at that question, my colleague, Luke Campagnola, created a model of synaptic dynamics. And I won't go into details of his model, but to say the least, he stuffed all of those properties into this model. It created a high dimensional space that then we could reduce down using a UMAP reduction. And so in this sort of butterfly shape, which is the two main UMAP axes, we have individual connections here as each dot, where adjacent dots are going to be connections that are similar to that one, whereas dots that are over here are connections that are very dissimilar to that one. And so how cells or connections cluster in this sort of space gives an indication of how similar or dissimilar those synapses are to one another. And so with this in hand, we can see how these relationships compare or relate to other things that we're interested in. And so if we first look at connection type, we see that the two main wings of this butterfly seem to be correlated with the two main classes of excitatory and inhibitory connections. So that's kind of cool, not super unexpected, and maybe a little bit disappointing that we don't see any fewer clusters in these data, beyond just excitatory and inhibitory cells. We didn't really need this model to understand the differences between those two. But there are some interesting patterns in here if we look at gradations across this space. And so again, to go back to this synaptic variance measure, if we colorize this plot by variance, we do see a striking pattern. In both wings of this butterfly, we see that along the edges lies a low variance regime, and then it kind of slowly transitions into a high variance regime here on the inner part of the wings. And to take this a step further, if we now color these dots by another metric, in this case for excitatory connections, the postsynaptic cell type, we again see that the colors kind of gradually change along this same axis dimension as the variance does. And so we have human excitatory connections on this outer low variance edge, transitioning to SST interneuron connections from the mouse in this high variance regime. Conversely, if we do something similar on the inhibitory wing, in this case coloring by the presynaptic cell type, we again see a gradation of color in the same axis direction. And here, parvalbumin interneuron connections are in this outer low variance space, and again, somatostatin SST interneuron connections in this high variance space. And so this is just one example of how these types of properties are graded across this high dimensional space. And there's a lot more that we explored that I don't have time to go into today, but fortunately, we just published a paper in Science, and you can get much more information and see a lot more data there. If you're interested in learning more about how we did our experiments, and you want to get your hands on the data and play with it yourself, we have a website that you can go to and get all of that information. And if you just want to talk to us more about multi-patch, about how we analyze this data or interpret this data, there's a community forum chat that the Allen Institute for Brain Science hosts, and you can reach us there. So I want to, thank everybody that was involved in this project. This kind of undertaking is not just for one or a few people to do, and it was a huge team effort. I'm part of the synaptic physiology group, which was the core group that undertook this project. And then we relied on many, many other teams within the Allen Institute to procure our animals and process the tissue and get the data up online so that you can all access it, and our communications department for hosting the forum and the websites and these sorts of things. So the Allen Institute is a huge team effort that allows these large scale experiments to really take off and be productive and useful for the community. And so with that, I want to thank everybody else at the Allen Institute and our founder, Paul Allen, for his vision, encouragement, and support. And I'm happy to take any questions.
Excellent. Thanks so much, Steph. That was a really great talk. So Max has come back on screen, and I see we have a few questions already. So please do, feel free to put your questions in the Q&A section for Max and Steph while we have them here. It's a good opportunity to ask them any burning questions you have. We've got 10 minutes or so. So we'll kick off with the first one from Jerome Cole. Sorry if I have pronounced that incorrectly, but are you more likely to pick up electrical noise when working with such a large number of manipulators? Max or Steph, do you guys want to?
I mean, it certainly doesn't help. The more electrical devices you put in there, the more electrical loops you can potentially create. So the shields helped a lot, and practically eliminated any electrical crosstalk noise that we experienced, and it was a super simple solution. So that, along with just making sure that every other thing on your rig is grounded, should solve most problems. Our rigs also had grounded sides, and we also would lower a curtain in front of the rig once all of the cells were patched and we were ready to record the experiment, to really eliminate wind or other noise coming in from that side. So if everything is grounded properly, it should be okay. That being said, we spent a long time trying to source noise and figure out where it was coming from and eliminate it. So it's always a problem, but once you find it, you can control it.
So it can be done, but it's not that easy.
It took us a long time. It wasn't from day one.
Thanks for that. That's great. And from Philippa Timmins, we have, do you always have to use a system with the MMTP staging? How flexible is the setup?
Yeah, no, you don't need to always use the MMTP. The system is quite flexible, and we offer a lot of different options. So depending on what you prefer as a patcher, you can either have a few different stage options, or if you prefer the microscope to move, you can have that as well. And all of that will work well with the LinLab software.
Excellent. Thanks, Max. Question for Steph from Dongil Kion. What acquisition software do you use? Do you your own one or a commercial one?
So it is our own, but it's also available for others to use. It's called MES, M-I-E-S. It's an acronym that stands for something I can't remember right now, probably multi something, something, something. And it is available on on the Allen Institute's GitHub website. You can download it, use it for yourself. My manager, Tim Jarsky, was the developer of MES, and so it's a great software. They're continuously working on it to improve automation, and yeah, happy to, if you look for that, if you have any questions, reach out to us. We can hook you up with using MES.
MES, okay, great.
It's run by Igor. Igor is the underlying acquisition for that.
Okay, MES by Igor. Okay, I'll try and post that in the chat as well. I have a question here from Sarazat Cavrel, and it's for Max. Hello, thank you for a great talk. I was wondering if multi-patching setup allow AP recording from granule cells in the cerebellum. Why, I ask, because granule cells are the smallest ones.
I don't see any reason, right now that it wouldn't work, right? If you're able to patch one cell, you should be able to patch multiple cells. So I don't think, just considering the size being an issue, I don't see why you wouldn't be able to.
To just go for it with those.
That's right. Yeah.
Yeah. Excellent. Thanks, Max. And from Elle, hello, thank you for a great talk. I was wondering if multi-patching setup. Oh, is that the one I just read? Oh, sorry, that was the one I've just read. So that's already been answered. I can't see any other open questions at the moment. If anyone has any more questions for Max or Steph, please do post them in the chat or in the Q&A section. We've got a few minutes left, and if something comes to your mind afterwards, then you can feel free to email me, louise.tratt@bna.org.uk, and I can pass your questions on to Max and Steph. So we just have a comment here. Can you see that, Max, from Josiane? Would you like to comment on? Do you want to read that and comment on that?
I was thinking maybe Steph might be able to.
Yeah. So during the installation of the octopatch rig, do you suggest adding one electrode at a time or another approach? So we took our approach was we would load all 8 electrodes up, bring them in to a coarse view of the microscope, 4 times objective, find all of the electrodes. Then we would go down to our tissue at 40 times. Again, bring all of the electrodes in, make sure that we found them all. If there was any drift, we could correct that and really lock down the position of each of those pipettes. And then we would go, our electrodes are kind of all sitting just over the top of the liquid, the ACSF in the chamber. And so they're not super far away, but they're not directly in the tissue yet, because you want to limit that. So from there, we would bring in one at a time, seal onto the cell, as Max mentioned, bring the next one in, seal, bring the next one in, seal, and so on and so forth. In reality, Max mentioned it's ideal to seal them all and then break in, which that is ideal. In reality, it doesn't happen all the time. Often, as you're bringing more and more electrodes in, that movement, little bit of residual movement will kind of cause your sealed cells to break in, but usually that's okay. So yes, we would kind of do it one at a time, but we tried to bring things in to a little bit closer so that things weren't traveling at huge distances to get to the cell.
Yeah, that makes sense. Thanks, Steph. Got another question from Isabella Savin. In addition to short-term plasticity, can you look at things like long-term potentiation, depression with patch clamping?
Yes.
Yeah.
No problems. Yeah.
There you go. No problems. Easy as that.
Well, yeah. That might be hard with multi-patch, but just because the experiments don't tend to last long enough for those sorts of protocols. But go for it. See what happens.
Brilliant. Okay, if anyone else has any questions, we've got 3 more minutes. If anyone's got anything to add. Isabella says, thank you. Sorry, I'm a beginner here. No problem at all, Isabella. Of course not.
No problem at all, yeah.
No, that's what we're all here for, to learn.
We were all beginners.
So yeah, exactly. And a few people saying thank you for a great presentation, and Steph, congratulations on the paper. Fantastic perseverance. Excellent. Okay, so I don't think we have any more open questions. If you do have a burning question, feel free to email me, louise.tratt@bna.org.uk. I'll pass them on to Steph and Max, who I'm sure will be happy to take your questions. So I think all we need to say is thanks for joining us again today. This will be available on demand to watch afterwards. So if you want to tell your peers and colleagues, then we'll ensure we send the link to you. So thanks so much for your time, Steph and Max. It's been a great webinar. And thanks to everyone for joining us. And we'll see you again soon. Thank you. Bye.
Thank you. Bye-bye.
Bye. Bye.